Good morning, AP Bio. Today we're going to talk some more about DNA, and we're going to start getting into the most important function of DNA, which is the fact that it carries the information, it carries the genes that code for proteins and traits and how to build your body and make your body function. First, I wanted to start by just looking a little bit at these diagrams. This is actually an idea that we will come back to again later, but it's important to think about today. So far, we have been focusing mainly on the double helix structure of DNA. We talked about all the details of that double helix structure. And we also talked about how that double helix structure allows DNA to replicate itself, to separate down the center of that ladder and then rebuild the two complementary sides. DNA doesn't exist by itself, though, really ever. And the main reason is because DNA is such a long molecule, it literally would not fit in our cells without getting tangled up if it wasn't organized in some way. And so in these pictures, it's two different ways to visualize the same thing. It shows you how that double helix of DNA, which is up here at the top on the right, is wrapped around these types of proteins called histones. That's these purple circles here. And it wraps in bundles. Those histones kind of collect themselves into these little groupings of 8 histones with DNA coiled around it. When the DNA is in that coil up section is called a nucleosome. You can see that here. When it's coiled up like that, this is referred to sometimes as beads on a string because that's kind of what that looks like. This is actually a photograph of DNA in this state. Another name for DNA like this, over on this left picture, it's called chromatin. This number 2 right here in the middle, that beads on a string where it's wound around those histones, but it's still fairly loose, is called chromatin. And this is how DNA exists most of the time in your cells, as this beads on a string. The only time that it coils up really tightly into these supercoils and chromosomes is when it becomes visible during cell division, which we'll talk about eventually. But for today, I wanted you to take a look at that chromatin structure, this beads on a string kind of structure. Because remember, the function of DNA, like we said, is to carry the genes that code for traits. And so those genes need to be accessible so your cell can actually get in there and copy the information to build the proteins it needs. And you'll notice here some parts of the DNA, the parts that are actually wound up really tight around those histone proteins, are not very accessible. The proteins can't get in there and separate that DNA and read the code. The part that's left accessible is kind of this in-between piece, the string part of the beads on a string. Those little in-between sections are the most accessible. And so this is the way that the DNA winds up around the histone proteins is actually really important. And it's kind of part of what makes different cells in your body able to make different proteins. And that's the part that we'll come back to a little bit later. But if you think about it, your DNA is identical in your skin cells, your brain cells, your muscle cells, your bone cells. They all have the same set of DNA, but they each make very different proteins depending on their function. And that's because the part of the DNA that is left open and available between these beads is a little bit different in each cell. And so different genes are available to be read by the cell. The next part of this unit, we are going to start talking about how those genes are read and made into proteins. And this is a process called gene expression. If you're following along in your note packet, this is where we are kind of skipping ahead. Remember, I rearranged some things. The middle section of your notes we will come back to in about a week and a half. Today, skip ahead until you find this slide. We're going to move into chapter 14, which is about how we get from a gene to a protein. So this is something in biology that is called the central dogma of molecular biology. And I put the definition of dogma up there just so that you kind of understand what this means. A dogma is like a central or core belief or tenet. You often hear that word in relation to religion, like the dogma of a religion is like the main core belief. And obviously this is science. We're not talking about belief. But that means this phrase, DNA makes RNA and RNA makes protein, that's been referred to as the central dogma because it's really central and foundational to the study of biology. So take a minute and think about this. This is something you learned about in 10th grade. What do you remember about going from DNA to RNA to protein? So this is the process that we will be talking about in a lot more detail over about the next week, is this process of going from DNA to RNA to protein. This is the main flow of information in living things. And so this process, which we sometimes call protein synthesis, is divided into two main steps: transcription and translation. You might have talked about these when you were kind of remembering how this process works. Transcription is basically this first arrow here. It's going from DNA to RNA. So transcription is the process where the DNA is copied to make a messenger RNA, that's that mRNA, and it happens in the nucleus because that's where the DNA is. So that's just a real quick review of transcription. We'll get into more details in a couple days. The second half of the process is translation. So translation is kind of the second arrow going from RNA to protein. So that's when that messenger RNA is then used to build the protein. And that's the part where transfer RNAs, those tRNAs, help out, and it happens at a ribosome, which is made of another type of RNA called rRNA, ribosomal RNA. So you can see there's these three different types of RNA that are involved in this process. And RNA, of course, is another nucleic acid, which means it's still made of nucleotides. Those nucleotides still contain a sugar, a phosphate, and a base. The sugar and phosphate bond to the next nucleotide using dehydration reactions to form that phosphodiester linkage to form a big chain of RNA nucleotides. So the basic structure of RNA is the same. But there are some important differences. Here are some of the differences that you might remember. RNA is usually single-stranded. DNA is a double-stranded molecule. RNA contains a different sugar instead of deoxyribose. That's what the D stands for. The sugar in the nucleotides of RNA is ribose instead. It contains a different base. This is one of the main differences. There's no thymine, there's no T in RNA molecules. Instead, there's that base called uracil. And RNA is also able to leave the nucleus. It can go out and carry that message throughout the cell, and transfer RNA goes out and works in the cytoplasm. DNA cannot. It stays in the nucleus except during the process of cell division when it has to move around to go to new cells. RNA is also often a temporary molecule, like messenger RNA is built, it goes out to the ribosome, it's used as long as your cell needs to build that protein, but then that molecule of RNA is degraded and breaks down, and those nucleotides are reused. DNA, however, is kind of the permanent copy of those instructions in the nucleus of each of your cells. So this process of using DNA, those genes, to make proteins, or sometimes to make RNAs, we'll talk about that later, is called gene expression. Okay, so you'll hear that phrase a lot. It's the title of this chapter in your book. Gene expression means taking the information in the DNA and using it to build an organism or make an organism function. So expressing those genes. So we're going to talk for a minute about the word gene. This is a concept in biology where our understanding has evolved and changed over the course of time. Mendel was the first one who came up with the idea of a gene when he did those experiments with pea plants. And he just realized that there was some piece of information that he called a gene that determined a characteristic or a phenotype, like whether the peas were yellow or green, and whether they had round seeds or little seeds, those phenotypes. So he figured there was one gene that determined each of those traits. That was kind of our first definition. As scientists started learning what genes were, T.H. Morgan did some experiments with fruit flies and realized that genes were located on chromosomes because he could see the chromosomes kind of moving around as egg and sperm cells were made through that process of meiosis. And so he realized, okay, a gene must be a section of a chromosome. And then scientists figured out that within a chromosome, it was the DNA that actually held the information. So they said, okay, it's a sequence of those A's, T's, C's, and G's. That's what a gene is. It's a section of that code. And then they realized that each of those genes made one specific enzyme. But then as we learned more about enzymes and proteins, we realized some of them actually have a couple different pieces made by different genes that then come and connect together in that tertiary protein structure. And so then we said, okay, one gene builds one polypeptide, one protein chain. But even that's not totally accurate because All types of RNA, like the transfer RNAs that come in later in protein synthesis, are also coded for by parts of your DNA. So we're going to come back to this idea and kind of talk about our current understanding of what a gene is after we've gone through part of this unit. But we've kind of slowly been refining our understanding of this word. So as we talk about this process of protein synthesis, we're really talking about gene expression and how we take that information in DNA and make it into something meaningful within an organism that does something. We are going to be doing a modeling activity as we learn about protein synthesis. And so today, your task is to do all of the setup for this activity. So I will need you here to grab the packet that I sent home with you. So you should have this packet that's got this little note on the front. It had this worksheet on the front, which we have already done. And then the rest of it contains a whole bunch of colored pages. This is the materials we'll be using for the modeling activity. So you need to grab this packet. One thing I do want to point out right away, somewhere in your packet, you have a little tiny piece of white paper like this. You don't need to be able to see what's on it. There you go. As you start taking apart these sheets, please make sure you don't lose this. We will need that as part of this modeling activity on Friday. So please make sure not to lose this. We are going to start cutting this packet apart today. And it'll be really important that you keep track of all the pieces. So you might want some paper clips or some baggies. If you happen to have a shelf or a table or a drawer where you can keep this activity so it's easily accessible, we're going to be working on this over about the next 4 class periods. So it's going to take a while. So here are the steps we're going to do today. First, you are going to take the gray piece of paper. This one in here is your DNA strand. And the first thing you need to do is just take it and cut it apart into strips. There are 4 strips on that paper. So cut those 4 strips apart. And then when you assemble your DNA, this is really important. So please don't do this wrong or the whole rest of the activity will be wrong. On each of those strips, I'm going to make my picture bigger again so I can show you this. There is a little number in the upper left-hand corner here. You can see this one is number 1. You need to tape these together in order. So that's my number 1 up at the top. If you go along, the next one has to be number 2, and the number should be right side up, and they should all be in the top left side. And I didn't tape the next one on, but the next one would be a number 3 right here. So it's really important that you tape those together in the correct order, kind of like this picture down here shows. And so when you're done, you should have just one long strand of DNA. This is just half of the double helix, the DNA double helix. We are not making the other side because we are only concerned with the template strand that contains the gene that we are going to be using to make a protein. So yes, in a real DNA strand, there would be another side down here. You don't need to worry about that for this. We're just having one big long strand of DNA. The second thing that you're going to do today is you are going to cut apart your RNA nucleotides just to save yourself some time tomorrow. The next 4 pages in your packet look like this. You've got C's, G's, A's, and U's, and they're all different colors. You do not need all of them. Please don't cut them all apart because I'd actually like you to bring the extras back to school, and I will reuse them when we do this activity next year, so we're not wasting so much paper. It was just much easier for me to send a whole page of them home with you, but you only need 15 each of the C's, G's, and U's, and you need about 20 of the A's. So cut those out. When you cut them out, they don't need to be totally perfect. Most people like to have the end where the base is, where the C is, cut pretty closely to where it goes, because obviously this is the part that will be matching up with your DNA. But you want to cut out each individual base like this, and keep them all together. So either paper clip all of your C's, all of your A's, whatever, or put them in little baggies or something so you don't lose them. Okay. So cut those all apart. And like I said, save the extra. The other, you should only use probably half of each of these pieces of paper. Save the other half, put them with your algae bead lab. So you have a pile of all your stuff that you'll be bringing back to school in a couple weeks. The last thing you're going to be doing today is making yourself an enzyme. Because, of course, there's an enzyme that helps to attach the RNA nucleotides during the process of transcription. You can just grab whatever piece of scratch paper you have around. If you want to make it pretty and fancy out of construction paper or something, that's fine. I had a little notepad of pink paper, so here is my RNA polymerase that I cut out. The shape doesn't really matter. It should be roughly the size of your fist, just so you have something about the right size. And write the word RNA polymerase on it. It could even just be a little sticky note that you use as your enzyme. That's totally fine. You just want to have something to represent that part of this process so that you remember that it happens. Please keep all of these pieces together. We will be starting the actual process of protein synthesis tomorrow, and you'll need all of these pieces along with some tape tomorrow to do this. For today, you have mastering biology homework that is due tonight. So if you haven't finished that, please make sure you finish that up. And I hope you have a great rest of your day.